Hello and welcome to all my dear lovely students. I hope you all are doing fine and must be studying well at home. So, today we are going to start the final lecture of the chapter Sexual Reproduction in Flowering Plants. This is going to be lecture number 7. I hope you all have gone through the first 6 lectures and have understood the chapter very well. Plus, must be also solving given DPPs on time. If you are doing this, then surely students, I can guarantee that you will understand the concept of this chapter very well. Now, today we are going to discuss the end of the chapter and we are going to discuss about some more post-fertilization changes, that is formation of seed from ovule and formation of fruit from ovary wall. Apart from this, there is one more additional topic by the name Apomixis. Apomixis is a type of asexual reproduction exhibited by plants apart from vegetative propagation. Now, in my last class beta, I have already told you the two most important post-fertilization event, that is endosperm development and embryo development in both monocot and dicot plants. So now let's start first with formation of seed as one of the post-fertilization changes. Now beta, when inside the embryo sac fertilization takes place, I mean to say formation of diploid cell zygote takes place, this induces the ovule, the integumented megasporangium, to transform into seed. So how will you define seed? What is seed? Seed is basically beta matured ovule, right? Now, if I want to define structurally what seed comprises of, so in order to understand that what seed comprises of, first you need to understand the whole structure of ovule. We have already done, but chalo, let's take a quick outlook. So this is the most common type of ovule, which is anatropous, right? These are the chalaza, the micropilar opening. These are the two integuments, the outer and inner integument. This is outer integument. There's a purpose beta why I'm drawing everything. And then this is your inner integument. Correct? Now, inside it encloses the female gametophyte, which is embryo sac. Now, after fertilization, there is formation of zygote. And apart from zygote, there's a triploid tissue produced due to triple fusion, which is endosperm. Now, soon after fertilization, beta, antipodals, they degenerate. Synergit's work is done. Remaining nucellus also get degenerated. In some, it may remain persistent. That we will discuss later. The two integuments, they will develop into seed coat. Now, these two integuments will develop into seed coat, which will enclose the zygote and endosperm inside it. So now this is my seed coat, which is actually double layered. Outer integument develop into outer hard testa and inner integument develops into inner soft tegmen. And both these together, testa and tegmen, are known as seed coat that develops from the integument of the ovule, which encloses the diploid cell zygote, which later develops into embryo. Plus, they can also have a triploid tissue called endosperm. All these topics beta you have already studied in class 11th while you were studying seed and fruit in the chapter morphology. So here I'm just going to give you a quick overlook of the structure of seed. So structurally, seed consists of 3 parts. Seed coat, which develops from the integument. that encloses the diploid embryo. This embryo is basically the progenitor of the new generation, plus the third triploid tissue called endosperm. However, presence or absence of endosperm is not 100% characteristic of seeds. Dicots, they don't have endosperm persistent in them because the complete endosperm is consumed in embryo formation. However, in monocot seeds, endosperm persist because there in embryo development, the endosperm is not completely utilized. So on this basis, on the basis of the presence of endosperm, we basically have two types of seed. Chalo, let's discuss. But for that, I need to, I think, add a page. Okay, so now first we are going to see types of seed. Number A is beta endospermic, also known as albuminous seeds. The seeds in which endosperm is persistent because it is not consumed in the development of embryo. Majority of the monocot seeds, except one or two like orchids, they are endospermic seeds. So what are endospermic seeds? Seeds in which endosperm persist. Example in monocots except orchid. Right? Opposite to them are non-endospermic beta, also called as Ex-Albuminous seeds. Now, these are the seeds of dicot where endosperm is not there in the seed because it is already consumed by the zygote for forming embryo. In my last lecture, I have already shown you that how suspensor is formed, which is responsible for absorbing the nutrition from the endosperm. So what will you write over here? Seeds in which no endosperm persist. No endosperm persist in their seed. Example in dicots. Do we know any exception? Yes, except like castor. Now, castor is a dicot plant, but still its seeds, they have persistent endosperm which is oily in nature. And yes, why it is persistent? Because the zygote development into embryo did not consume endosperm. Understood the two types of seeds? One more category of seed we know is perispermic seeds. Beta, normally what happens? Nucellus is either consumed up completely, or even if it persists, then it gets degenerated soon after fertilization. However, in black pepper or beet, in the seeds, some amount of nucellus may remain persistent. Such seeds in which nucellus is also there, remnants of nucellus are also present, then it is called as perispermic seeds. So seeds in which nucellus is also persistent. Like for example, in black pepper, that is piper, beet, etc. Such seeds are perispermic seeds. Understood till here? Yes or no? Okay. Now, beta in NCERT, one point is given on seed viability. You know, see, now suppose seed is produced. Okay, fertilization is done, fruit is produced, inside the fruit seed is produced. Now you eat the fruit, you throw the seed away. Okay, that seed is enclosing the embryo. However, now if suppose outside conditions are not favorable for the germination of the seed, then will the seed let its embryo develop? Will the seed let its embryo to form plumule, radicle and cotyledons? No. In that case, seed is going to wait for the onset of favorable condition. Once the favorable conditions beta they resume, then only the seed is going to allow its embryo to develop. Till then, the time lag from the time of seed formation till its germination is known as seed dormancy. So what is seed dormancy? The time period for which seed does not allow its embryo to develop because outside conditions are not favorable. The period of seed dormancy is variable. The period of seed dormancy is variable. It can last for months, years or many years. So next topic is seed dormancy. Now what is seed dormancy? The time period for which seeds does not allow embryo to develop. And what is the reason? Due to unfavorable conditions. Right? Now, seed dormancy is highly variable. Like for example, 2 are quoted in your NCERT, which you have to remember. One of the seed, which is about 10,000 years old and still it's viable, is extracted from Arctic region and the name is Lupinus arcticus. So Lupinus seed has been extracted from Arctic region and its dormancy is for about 10,000 years. It's viable since 10,000 years. Can you see the dormancy range of this Lupinus arcticus? Similarly, there's a king called King Herodes. From his palace, also one of the seed was extracted of date palm, Phoenix dactylifera, whose dormancy ranged for up to 2,000 years. So next is extracted from King Herodes' palace. The dormancy range is for 2,000 years and they are still viable even after this period of time. So I hope you have understood the seed. Now apart from seed, if it is an angiospermic plant, then around ovule there is one more covering by the name ovary wall, which then transforms into fruit. Ovary wall transforms into pericarp and ovary transforms into fruit. So next post-fertilization event is formation of fruit. So what is fruit? Basically it is ripened ovary or fertilized ovary. Fruit beta. Now around ovary there is ovary wall like this. So this is your suppose ovule. This is ovary. And this is ovary wall. Now this ovary wall is going to transform into the wall of the fruit, which is called as pericarp. So this is also one of the post-fertilization modification, that means transformation of ovary wall into pericarp, which is actually the wall of the fruit. Correct? Now, there can be three types of fruit, beta. True fruit, false fruit, and parthenocarpic fruit. See, details already you have done in class 11th chapter, morphology chapter. Here we are just going to discuss the little bit portion which is important from the point of view of this chapter. True fruit, fruit which develops only from ovary. Majority of the fruit, 99% of the fruit are true because they develop only from the ovary. However, in case of apple, strawberry, cashews, along with ovary, if some other floral part also contribute like thalamus, then that is known as false fruit. So what is false fruit? Fruit in which along with ovary, other floral part like thalamus also contribute. So it becomes false fruit, like for example apple, cashew, strawberry, etc. However, in true and false, both fertilization is taking place. The third category of fruit is parthenocarpic, where fruit is formed without fertilization. Ovules are not transforming into seed. There is no formation of zygote taking place. There is no fertilization taking place. But still, simply ovary wall is transforming into pericarp. There is only fruit formation without seed formation. Such type of fruit is known as parthenocarpic fruit. So what are parthenocarpic fruits, beta? Fruits which develops without fertilization. Fruits which develop without fertilization. Such fruits are seedless, like bananas, grapes, papayas. They are parthenocarpic fruits. Such fruits are seedless, like your banana and papaya. Grapes also. These are basically parthenocarpic fruits where no ovule are transforming into seed, no zygote formation is taking place. It's only the ovary wall which is transforming into fruit wall. It's only ovary transforming into fruit. So they are going to be seedless, like you see in case of banana, you see in case of grapes. Right? Understood? Now, many more fruits are there, fleshy fruits, dry fruits, that you have already done in your class 11th chapter, morphology. For this chapter, the only important things were to know about true, false and parthenocarpic. I hope these two post-fertilization changes are also clear to each and every one of you. Now, we are going to start the very important and the last topic of the chapter, Apomixis. Apomixis, beta, it is actually a special type of asexual reproduction where you are going to see seed formation without fertilization. We say that we can make fruit. You have seen in parthenocarpy, fruit is being produced without fertilization, but seeds cannot be produced. However, apomixis is a type of asexual reproduction in which without fertilization, we will not only produce fruit, but we will also produce seeds inside them. So this type of asexual reproduction, which mimics sexual reproduction, is known as apomixis. It was first reported by Leeuwenhoek in citrus fruits. Correct? Naturally, apomictic genes are found in the most advanced families of dicots and monocots, that is Asteraceae and Poaceae respectively. Apomixis can be done by two ways. One is known as polyembryony and second is known as recurrent agamospermy. So first I am going to write these points and then I will explain you the details of it. What is apomixis? It is a special type of asexual reproduction, beta, which mimics sexual reproduction. How? Now, how will you say this? As fruits plus seeds both are produced without zygote formation, without fertilization. Both are produced without fertilization. Right? Next point. It is naturally observed in the genes for apomixis. They are naturally found in the two most advanced families, that is Poaceae, commonly called as grasses, and Asteraceae. Right? First time apomixis was reported by Leeuwenhoek in citrus fruits. Now there are two ways to perform apomixis. One is called as polyembryony and second is called as recurrent agamospermy. Now let's see both the ways of apomixis one by one. First we are going to start with polyembryony, also called as sporophytic budding. So Adventive, the full name is Adventive polyembryony, which can also be called as sporophytic budding. It was observed by Leeuwenhoek, and some examples of this you see in case of citrus, you see in case of mango, you see in case of opuntia. Okay? Now, what is this all about? See, what happens, beta? This is my ovule. Let me draw a proper natural ovule. This is my anatropous ovule having two integuments. Correct? This region is micropyle. This is the embryo sac, three antipodals, central cell, egg, synergids. Okay? Now, these are the integuments and there's remaining lot amount of nucellus present. Now, what happens in citrus and mango, beta? There is natural fertilization taking place. Leeuwenhoek said that in citrus or mango, pollen tube is coming, bringing the male gametes, delivering them inside. Fertilization is taking place. Zygote is forming naturally. But apart from natural fertilization, apart from natural diploid cell, zygote that develops into embryo, apart from this, some of the integument cells and some of the nucellus cell, they pop up, they start growing in size. Now they are diploid in nature, so this is natural embryo. Natural sexual embryo, sexual diploid embryo in case of citrus or mango. But apart from this, inside the embryo sac, some more diploid cells are pushed inside, which have arisen from the sporophytic budding. Sporophytic means diploid. Budding means growth of diploid cell. because of which these cells of integument and nucellus can also behave as embryo. Now, in one sac, there are more than one embryo, one natural sexual embryo and some more asexual embryo. So, this condition is known as polyembryony. Many more embryos in a embryo sac. Correct? So, what is this? What is polyembryony? What is sporophytic budding? Apart from natural embryo, some of the sporophytic cells. Sporophytic cells means cells of nucellus or integument. That is nucellus or integument grows in size and push themselves inside the female gametophyte, which is your embryo sac, giving rise to a condition called polyembryony. Now beta, simply what do you mean by embryo? Embryo simply means group of diploid cells which are capable of further division. Now this embryo has developed from single diploid cell, which is zygote. Now this is the sexual one which is produced after the fertilization. But all these diploid cells which you see due to budding, due to sporophytic budding, they are also diploid in nature. They are also actively dividing. So can't they behave as zygote? What is zygote? Zygote is nothing but a single diploid cell which is capable of dividing. Now, that may be sexual, they may not be sexual. Naturally, zygote is a sexual term which is produced after fertilization. However, if the same property is exhibited by any other cell, then it can also behave as zygote even if it is not produced by fertilization. So same thing is happening with these cells. The sporophytic cells of nucellus and integument, they pop up. They grow in size. They regain their capacity of division. They are diploid in nature. They push them inside the embryo sac. Now each this diploid cell can divide and can give rise to multi-cell diploid structure, that is embryo only. So like if this embryo can develop into new organism, then each cell embryo can also develop into new organism. The only difference will be the organism developed from sexual embryo will not be identical to the parent. However, the new offsprings developed due to these embryos, they will be clones of each other as well as with their parent. So they are asexual embryos giving rise to clones. This is sexual embryo giving rise to new organism, variable different from the parent. Now this much happened. So this ovule will then transform into seed and then ovary wall will transform into fruit. So now in citrus and mangoes, what you see? You see some normal embryos and some asexual embryos also. So if you get in lemon, if you have suppose 10 seeds, so in those 10 seeds inside the embryo, now if you suppose plant those 10 seeds, so there may be 4 to 5 such seeds having asexual embryo. So now the new plant arising from those seeds will be exactly identical to the parent. Understanding? So this is apomixis. See, for producing these embryos, for these asexual embryos, no sexual reproduction is done, but still seed is formed, but still fruit is formed. So this is apomixis. Now another way is beta recurrent agamospermy. Now, in recurrent agamospermy, as you see in case of maybe apple, right? Agamospermy. In this case, at least in case of citrus and mango, you saw at least one natural embryo, okay, and remaining were asexual embryos. However, in recurrent agamospermy, there is not even one natural embryo. All the embryos, they are asexual, they are produced without fertilization. Let's see how. What happens in case of recurrent agamospermy, beta? There is, megaspore mother cell. What happens in natural condition? This diploid megaspore mother cell that develops from one of the nucellus cell, it undergoes reduction division, it undergoes meiosis, 4 haploid megaspores are produced, out of which in majority of the cases 3 gets degenerated. The one functional haploid megaspore then undergoes free nuclear division to form the complete haploid female embryo sac, that is female gametophyte. However, in some cases, these megaspore mother cell, they do not undergo meiosis, rather they undergo mitosis. And they will not produce haploid megaspore, rather they will produce diploid megaspore. Now this diploid megaspore will further undergo mitotic division, equational cell division, to produce a diploid embryo sac. So normally embryo sacs are haploid, and each cell inside the embryo sac is haploid. But here the embryo sac is diploid, means all the cells, suppose there are antipodals, they will be diploid. Suppose there is egg, it will be already diploid. Now beta, if the female gamete is already diploid, have the capacity of division, can't it directly behave as zygote? Shall it wait for the male gamete to come and fertilize? Now, if male gamete is going to fertilize with this diploid egg, it is going to form a triploid cell, not a diploid, because egg is already diploid. It does not need the mechanism of fertilization. It itself can directly behave as zygote and can lead to the development of embryo even without fertilization. So such case where megaspore mother cell, instead of undergoing reduction division, if they undergo equational cell division, then they are going to form a diploid embryo sac, already having a diploid egg. In this diploid egg, there is no need of fertilization. This egg can directly behave as zygote, and hence there is no need of fertilization. Ovule will sense the ovule. Now this embryo sac is around the ovule. Now, because of this diploid cell already, ovule will get confused that, okay, diploid cell is there inside the embryo sac, I shall transform into seed. Even without knowing that beta no fertilization took place. This is already female gamete whose nature is diploid. So ovule won't come to know about it, only sensing that diploid cell is present. So ovule will transform into seed, thus giving signal to the ovary wall to transform into fruit wall as well. So here also no fertilization took place, still fruit is also formed, seed is also formed, and diploid embryo is also present. Now such seed, fruit you take, you eat, seed you plant it in the soil. Now, such seed is having apomictic embryo. So now this zygote is going to develop into beta. It is going to develop into apomictic embryo. Seed is going to enclose a apomictic embryo. Now, when you sow this seed in the soil, the embryo is going to develop having all the same properties, identical properties as that of parent. So that means you have replicated the qualities of the parent without sexual fertilization, without performing any fusion of gametes, without any fertilization event. Simply we have transferred the characters from the parent to the offspring through asexual method, and that is apomixis. So this embryo is going to carry all the property of the megaspore mother cell, which is a parent sporophytic cell. Understood? Yes or no? So can you imagine that how without fertilization even seeds are getting formed? How? Because even without fertilization there is formation of diploid cell inside the embryo sac that signals the ovule to transform into seed. So this is about apomixis. What is apomixis? It is a type of asexual reproduction which mimics sexual reproduction because fruit formation के साथ along with fruit formation there is also seed formation. Why seed is forming without fertilization? Because to form seed, to transform ovule, the prior requirement is formation of a diploid cell inside the embryo sac. And here you see how we have produced a diploid cell inside the embryo sac without fertilization. So ovule gets the signal, transforms into seed, transforms ovary wall into fruit. So the process is done. With this, we come to the end of the entire chapter, that is your sexual reproduction in flowering plants. Now I have some questions in front of you. I hope today class was very small. So just one question to practice. Do the practice. Do read the NCERT book, which is very important. And yes, DPP will be given to you. Solve these DPPs. Buy some new objective books so that you can solve more questions from this. Till then, see you. Bye-bye. Take care.